Okay. So hopefully you've seen the emails from me regarding the fourth exam you took on Monday and all of that. So according to my calculations, there's quite a few of you who can opt out of the final exam if you choose to. And the way to do that is just send me an email and say, I'm opting out. Okay, that's all you have to do. And then if you're like waffling and like, oh, I might want to try to boost my score, just be aware the final exam counts. So if you take it, it counts, good or bad. So if you don't get a chance to study for it and take it and you do poorly, it's going to count either way. So just keep that in mind. And then remember that the lab grades get added in, of course, to the overall class grade. So sometimes students forget that typically lab grades boost overall scores. Yes. Total points that you have, so you just take whatever that yeah, so I take your total, add 15 points, and I think the total lecture exam points is 367. Okay. Does that include the 5 points for that? Yes. Yeah, so I think it's 15 total with all 4 exams. And I saw another hand. Yes. So I just posted right before now. I just posted into EduCat in the very first section of EduCat where you go to click on the exams. There's a Word document there now, and it says final topics to be covered. I have part 1. Okay, so I've worked it all the way through to the light reactions. So that's part 1 of the semester, and then I will finish fleshing that out. So you'll see what's going to be on the second exam. So I thought if you have all of you have a way to get online, if you could pull up that document and look at it, and then I have my copy here, and I thought we could go through the slide sets and clarify any questions that you may have. But I think if you look at the Word document on the outline of what's to be covered, that will help you kind of orient through today. And then remember, Friday as well, we'll do another review session. So sacrificing 2 lectures for this. So let's make it count. And so the exam format will be multiple choice. There might be some fill in the blank. Like it would be like a 1-word answer or a 2-word answer, you know, that kind of thing. So those are more like definitions. So that's multiple choice, true, false. There might be some drag and drop. I haven't finished making the final exam yet, so I don't know. But there will be no essay. No essay. So you'll just see exam 5, final exam. That's the only thing you have to click on. You won't have 2 separate things to worry about. Okay. So right now the draft is around 90 points. I don't envision it going over 100. So it will be, I'm guessing, somewhere between 80 and 100 points. Usually when I make exams, I kind of overshoot and then I cull back. I start deleting things. That's usually how I work it. And it takes many days to work through this process for me. So I can give you more feedback, maybe even by Friday, or via email. I can give you some feedback. The final exam is Wednesday at noon in this classroom. Yeah, I'm thinking about options for that. The one problem I've encountered on that, well, one of the problems I've encountered on that is my final exam questions don't look like, they're not the same questions. And so I've had students not understand that. And so they just kind of like try to memorize, and then they're not thinking really through the material. They're just trying to memorize everything. And then they do poorly on the final exam. So I'm really trying to figure out a way to not have that happen, because it's just so sad when I see Yeah, the way I've been handling it is I would be very willing, even into next week, to work with you one-on-one to so outside of my office hours too, I have time available. So if you feel like you really want to go through the exams, please see me. That's how I'm handling it right now. I'm still trying to see. I've seen so many students over the years of teaching this class where they just try to memorize everything. And my exam questions, I don't take previous exam questions and put them on the final. It's not how I do it. I kind of make up a brand new, because what I'm doing, if each time you take an exam, it's probably similar in other classes too, but it's on, of course, on a smaller set of content, right? And so I can get more detailed in what I'm expecting you to know. But for the final, I kind of pull back a little bit, so there's not as much detail. So that means I have to word things differently. The answers have to be presented differently. And so it's really a different beast. However, I get what you say that, I'd like to see what I got wrong, what I got right. So I can kind of, yes, for sure. Let's set up times to do that. And I actually prefer to do that one-on-one because then I can work with each student to say, give tips and strategies for maximizing success for the final. So, yeah. So having said that, when I say I pull back for the final exam, I will try to make what I mean by that clear as we work through these slide sets today. So this was the first slide set that we did the very first week, looking at plant cell anatomy and tissues, architecture, so to speak. And so, I put on the outline, understanding cell wall anatomy, for sure. So, because that's very different. We don't have cell walls, plants do. They have not just one cell wall, but sometimes it depends on the cell. They'll have two cell walls. So knowing there's a primary cell wall, a secondary cell wall, their general composition, what makes a secondary cell wall unique with that added lignin, right? So that lignin is making a big difference on that. And then if a plant cell is destined to have a secondary cell wall, what happens to that cell? Do you remember? It dies. Okay. And so that, those are like big picture concepts. All right. So the idea that you have two different cell wall types, and then some plant cells are destined to die once they lay down that secondary cell wall. Chloroplast, of all the organelles, really, it's the chloroplast I'll focus on. I don't really envision talking about anything else in terms of organelles. So know the chloroplast anatomy. Oh, well, the vacuole. I can't forget the vacuole, sorry. So the vacuole, chloroplast and vacuole are the two things that you should review. And obviously the chloroplast, we spent a lot of time looking at it and then looking at what happens inside it. So that one's going to take front and center. And then plant tissues, we covered on this slide set as well. Oh, plasmodesmata. Did I put that on there? Yes, plasmodesmata, knowing that that's the channel linking neighboring cells. Okay. And then tissues, we have the dermal tissues, so the epidermis, and don't forget about the guard cells, of course, and the stomata, essentially, the guard cells surrounding the pore. That's in the dermal region. Ground tissue, just the parenchyma is usually what we talked about there. That's what I focused on. And then the leaves, of course, they had to throw more jargon at you with the leaf anatomy, looking at the palisade versus the spongy parenchyma. Mesophyll means the same thing as parenchyma. Okay, so don't get confused by that. So this slide set is fairly straightforward. It's basically anatomy, jargon that you need to brush up on. Are there questions on any of those? Okay. Next one, water, water. We looked at, so the initial slides here on water properties, I won't be asking it, again, I'm zooming out, so I won't be asking about specific heat or heat of vaporization and so on. But, so what are the most important topics here? Looking at Fick's first law of diffusion. No, you don't have to do calculations. We never did, so I'm not going to ask you to do that. But understanding the concept, right? You have delta C over delta X, so the concentration gradient that's really impacting what's going on with diffusion. And then with a Poisson, the French guy's name, Poisson equation, we looked at bulk flow, where we have a pressure gradient, looking at that idea. Whoops, that's not what I want. No, I can't do that with this. Okay, anyways. So Fick's first law, the Poisson equation is delta P over delta X. So we're looking at a pressure gradient due to bulk flow processes. Osmosis, know the definition for sure. That's a big deal in this class. And then the water potential equation. So you don't need to get into the details of how we figured out that it's easier to go with a water potential equation, but knowing, so slide 21 on my slide set, the overall water potential equation. What is it? Don't forget it's a plus sign, y psi sub p plus psi sub s, if I ask you to write it out or something. And then knowing what does each component stand for. So water potential, how are we feeling about that? I will be asking concepts like what happens if we have two neighboring cells or something like that, or if you drop a cell into a beaker. Do we need brushing up on that? You'll have some version of a story problem. You won't have to do essay, but you'll still have to think through it. So again, when you're looking at these concepts, whether it be an artificial cell that I talk about or a plant cell, you know, anytime you have a living cell, of course, there's a psi sub s component because there's stuff dissolved in the cell. And so that's messing with your psi sub s component. So knowing, you know, what each component stands for and how is it getting manipulated or shifted if you run through a story problem scenario. So water moves from less negative to more negative. Obviously, that's a big idea. So I'll be asking something where you can demonstrate you understand that. Less negative to more negative water potential. That's how water will move. So here you see psi sub W is negative 0.15 in the beaker, negative 0.25 cell. So less negative is the beaker, right? So you plop that cell in, and water will move from less negative water potential to more negative water potential. Well, as soon as you have that happening where there's a membrane involved, an intact membrane, you're going to get pressure against that membrane. Water's going to rush into the cell. It's going to create that Turner pressure. So now that messes with your psi sub p. Your psi sub p value will go up. So just be careful about the negative signs and positive signs. So when your psi sub p, in this case, in the cell, it's going to go up. It's going to be some positive number, okay? Now, when we get into transpiration, we think about a negative pressure or tension, but that's not the same thing as what we're talking about here. This is an intact cell, cell membrane and cell wall. That's going to definitely introduce a positive psi sub p into that part of into the cell. So if I present questions on the final, it will be, do you know what the overall equation is? Do you know what each part stands for? Do you know how water moves in relationship to this equation from less negative to more negative? So I'll be designing questions that will get at that, and then I'll probably have a story problem or, you know, a problem that you work through, where I present a scenario and then I'll say what's going to happen, and you'll have to pick or do a drag and drop or something. So do we have specific questions? Yeah. Correct. So if you start packing this full of a bunch of sugar for some reason, the cell gets jammed with a bunch of sugar, your size of F is going to get more negative. More negative. So since I have more control on how these questions are going to look, you're not going to be doing an essay. I'll probably be phrasing it that way. I'll say, would it go more negative or less negative? That's probably how I'll phrase it, because that's how I prefer you think about it, rather than higher, lower. That can get mixed up in your head. So, yep. Other questions on water potential concepts? Okay. We can come back to that if you have anything lingering, but let's look at transpiration next. So that's the movement of water through the plant body, but we can't ignore the soil. I will be asking on what's going on in the soil, as well as, so as the plant is taking up water from the soil, how is water moving through the soil? And so we talked about that idea, how it's really a tension gradient that's established in the soil. And so basically what's happening in the plant and in the soil, it's very analogous in terms of there's a tension gradient that we need to think about. And so although again, I'm not going to ask you to memorize the equation here, but just understanding this equation that your psi sub P, if water's under tension, that's a negative sign. Negative of the meniscus. Boy, that's a mouthful. So water in the xylem, it's under tension because tension is negative pressure. So this equation is trying to get you to think about that, and the idea that the radius has a big impact. And because it's in the denominator, if you decrease this, it makes the overall calculation to be a bigger number if you actually do the math. And so if you have a really small radius, you're going to get a really nice negative tension. And so that's why I put that equation there. If you have any math analytical ability, that play with the numbers a little bit to convince yourself. And so this next slide is also trying to get you to play with the numbers a little bit, convince yourself that as the radius of the curvature of the meniscus of the water, whether it's in the soil or up in the leaf, as that radius gets smaller and smaller, your overall tension gets more and more negative. It's a really nice thing to study on, I think, this slide. So you have to imagine that happening inside the leaf, right? You have the water coming out of the leaf vein, out of the xylem, and it's in its liquid form, and it starts to adhere to all the leaf spongy mesophyll cells, and then you end up with that air-water interface, right? And then as the water is evaporating and exiting out of the leaves, you have that liquid water evaporating into smaller and smaller crevices between the cells. So there goes your radius. Your radius is getting smaller and smaller and smaller. So that generates a nice negative tension. So it's all generated up in the leaves. So, well, assuming something's happened first. What has to happen first? In the leaves. Yes, stomata have to open. If that doesn't happen, you're not getting transpiration. So at night here, most of our adapted plants, there's C3 plants, they close their stomata at night to conserve water. So they're not doing transpiration at night. You have to have those guard cells, stomata open. Then you get air, water vapor evaporating out, exiting the leaf. And that will generate that tension because of the idea of the radius, the curvature of the meniscus gets smaller and smaller as all of that's happening. And that generates that really nice negative pressure tension. And so it's this tension gradient. You get this nice negative pressure, good tension up in the leaves, and then progressively less tension as you work your way down to the roots. And they can measure that. They have documented that. So questions on transpiration. And if there's anything on the outline, if you're not sure what I mean on the outline too, make sure to ask. That's your study guide. So don't be. Oh yeah. Apoplast and symplast ideas. So that's, when we're thinking about water movement into the plant from the soil. Is it on this? Yeah. So here we have the image of the root and cross section with the root hairs, and then you see the artist drew a couple of different pathways of how water might be taken up from the soil into the root tissue. It's correct the way it's drawn here, that it's the root hairs that do the lion's share of absorption. They're really, really good at that. Great surface area. And then they're saying, well, we could have the apoplast method where that's green here, where the water is just skirting in the cell wall space. There's no like obstructions that way. They can just skirt through until they reach the endodermis. Okay. That's limited there. But then transmembrane, they said the blue idea, which doesn't seem to really be happening in reality, but theoretically it can happen where you have water crossing membrane through the cell. That just seems like a lot of work. Or that last method, the symplast method, the black line is trying to show that, where once it gets into one cell, it just uses the plasmodesmata, that internal tunnel highway system from cell to cell to cell to cell. And then the only one that's really complicated at the endodermis is the apoplast. So in the cell wall, it runs into that Casparian strip, which is really that wax that's laid down in like a band in the cell wall. And so the water molecules bump into that, and that's it. They're like, okay, I can't go any further. So that forces them to pump in the water into the cells at that point and not stay in the cell wall space. So those three methods, but with water movement, it's really the idea of apoplast and symplast. Transmembrane, not so much. Any other questions on the transpiration slide set? Okay, transpiration, then phloem translocation is the next one. Let me pull that one up. Okay, so phloem translocation, still staying with the vascular tissue, but now we're focusing on sugar movement primarily, although there's more than just sugar in the phloem. But for this, we'll just kind of zoom in on the sugar. And so sucrose is the favored option for plants. Plants make all kinds of different sugars, but they choose sucrose. And so knowing the properties of sucrose is important. Why is that? The adaptation they evolved to use as their way of transmitting sugar through the plant. They pick sucrose. So just knowing a little bit about the properties of sucrose. So why sucrose? I have that slide there. Why sucrose? And then, of course, the jargon. Sugar is moving from source to sink, knowing the jargon and the concepts. The definition of what a source is versus what a sink. So slide 23 on this slide set is important. And then the pressure flow model that Dr. Mooch came up with is the favored hypothesis for that process. So again, with pressure flow model, it's right in the word, right in the phrase, pressure. We're looking at a pressure gradient. And it's a positive pressure that gets filled up in the source, a lower pressure in the sink, and so it's a pressure gradient. And so again, I need to design questions to make sure you are demonstrating that you understand it's a pressure gradient. Okay. And it's a positive pressure building up in the source and then a lower pressure in one of the sinks. And as long as you have that gradient, you'll get that movement from source to sink. But it's very dynamic. You know, sinks, as we know, we learned, sinks can become sources, perhaps. Baby leaf matures. Initially, it's a sink, and then it grows up. It's all grown up and can fend for itself. Then it becomes a source. So it's very dynamic. But the trick is, if there's a gradient, then you'll have phloem translocation occurring. So this graphic that has the phloem translocation, slide 30 here, is a good one to study off of course. So again, they're looking at water potential concepts in the two different regions, the source versus the sink. And so again, I'll be asking questions, you know, based on, well, what happens when you load sugar or if you unload sugar? And so here at the top in the source, top here, all the red balls are getting loaded in, all those sugars getting loaded into the Siv 2 element. And so understanding that that messes with your psi sub s, it makes it more negative. Your psi sub s becomes more negative as more sugar is loaded in. Well, that's going to drive your overall water potential more negative compared to a surrounding cell, especially the xylem right next door. Remember, it's in the vein, they're all clustered together. So you have a really negative water potential in the xylem. So it's really easy for water to start to react to this situation of the overall water potential in the phloem source getting more negative with all that sugar. Overall water potential gets more negative. Water will go from less negative to more negative, so it rushes into the source Siv tube element. And okay, well, that's going to mess with your psi sub p. Raise your psi sub p up. You've developed a nice pressure, positive pressure up in the source, typically a leaf. And then in the idea of the sink, I used a white potato tuber underground growing. And so, of course, you have to build the tuber, so you have to unload all the sugars and push it into the tuber. So that drives your psi sub s to become less negative. And you lose the sugar out of the Siv tube element, your psi sub s starts to become less negative. Which means overall your water potential gets less negative compared to the xylem right next door. And so water will leave, go from less negative to more negative. So you get this nice little circulating loop of water. But the upshot of that is once the water leaves the sink area, that drops your pressure. So now you've completed the story of a pressure gradient, source to sink. Questions on source to sink, water potential. I also have on the outline the polymer trapping model. And then the jargon, allocation and partitioning, keeping those two straight. So the definitions, knowing the definitions, and then that, I don't know, the polymer trapping model to me just sounds bizarre, but it works. Where is it? Polymer. Oh, there it is. So yeah, when they first came up with this, it sounds weird, but it works. So that idea that once the sugar is getting loaded in to the companion cell or the Siv tube element of the source, you have larger sugars getting made right there. And then if you look at the anatomy in that region in the source tissue, you actually see a difference in the anatomy where the plasma does not. They're bigger here, into the Siv tube element then what you see is just one of the mesophyll cells, much smaller of the plasmodesmata. So they've actually measured all that. So everything they've done to take measurements, they see the enzymes showing up there, different anatomy with the plasmodesmata. It's supporting this polymer trapping idea. So it's just a nice way to, this is thinking about the idea that these are all open channels of tunnels. So you would think if a bunch of nearby cells are loading sugar in one system, it gets lots of sugar, why won't it just feed back out just by the laws of diffusion? That's what was really making them scratch their heads. Because there's a lot of sugar that builds up in the storage development. Why doesn't it just feed back out? And so the polymer trapping model, they came up with that idea. They said, well, that would fix it. That would fix the problem. That would make sure that sugars it's a one-way movement from the leaf cell, the mesophyll leaf cell, parenchyma cell, one way, driving it toward the sieve tube element. It won't backflow. Anything else on phloem translocation? Whoops, what happened? Hang on. All right, did I see a hand somewhere? No. Allocation versus partitioning. So slide 57, 58 is showing those ideas. Okay, let's go into the next one, mineral nutrition. Okay, what happened to my, there it is. Mineral nutrition. Okay, so this one, as you see on the outline, I'm going to be zooming out a little bit on this. So really the only minerals that I want you to think about in any level of detail would be nitrogen, like what form do most plants prefer, and that's the nitrate version of nitrogen, NO3 minus. So that is important. And then there's some issues with phosphorus that even though you might get your soil tested and they say, oh yeah, you got a lot of phosphorus. The problem is phosphorus is very commonly precipitated out of solution in the soil. And so it's insoluble, precipitated out. Well, plants have a really hard time with that. They have a very hard time getting access to anything that's precipitated out. And so phosphorus is often really the one that's limiting the plant. It's the lack of available phosphorus. So things aren't really available to the plant until those minerals are dissolved in the soil water. That's then the case where the plant goes, yep, I can take that up. And so through the action of microbes help solubilize minerals, and then the cation exchange idea. So, and then the only other issues with phosphorus, and then issues with iron. So we looked at iron is technically a micronutrient, but there's some debate on that because it's needed so high. And again, iron can be a problem because there's different forms of iron, and so the Fe2 plus, that's more soluble, that's not so much an issue, but the Fe3 plus, the ferric form, is a problem. It's really hard for that to get dissolved in water, in the soil water, and plants have a hard time. And so that's why they have observed that there are some plants that have adapted where they make those phytosiderophores or some kind of chelating agent that helps them latch on to iron. So again, just this iron issue and the nitrogen and the phosphorus, those three are really what I want you to think about. And then just overall knowing these are macronutrients, these are micronutrients. Okay, so just being familiar with what's a macro versus a micro nutrient, which there's a table that shows all of that. And then just the big picture idea, like, what does chlorosis mean? What does necrosis mean? If a nutrient's immobile in the plant, well, then what would a plant look like if it starts to get a deficiency? Is it the older parts, the younger parts of the body of the plant? Usually you look at leaves. And so immobile versus mobile will give you different patterns of deficiency symptoms when you look at it with the naked eye. And that's very useful. Hopefully you'll make use of that through your whole life as you have plants in your life. You'll be able to tell if your house plants are getting issues with certain mineral deficiencies. Questions on any of that? It's fairly straightforward. Need to just brush up on those ideas. And then this slide set also got into mycorrhizae. And we only covered the ecto versus the endo. So those are the two I'll focus on. And, you know, there's some obvious major differences between ectos and endo in terms of how they interact with the root. What do they look like when they're interacting with the root? The surface of the root versus what's going on inside the root. Questions on the mycorrhizal fungi or they form the relationship? Need clarification on any of that? The terminology again, what's a mantle? That, you know, that's just a way of describing. So the terminology is important because you have to use those words to describe what you're seeing when you dig up these roots. Unfortunately, with the VAM fungi or the endomycorrhizal fungi, you don't have that nice mantle easily. So like, I know a lot of you did staining in lab. It's too bad you couldn't just dig up the root and go, yep, there's the mantle. That's what I chose to do for my PhD because I just like, oh, I don't want to stain all these roots. So especially when you work on a PhD project, it's massive. So it's like I'd be there for months staining and interpreting. The mantle, that's easy to pick out with the naked eye. So the terminology will be important. And then just, you know, what's the characteristic of the ecto versus the endo, not getting them mixed up, not confusing the two. Questions? Is your brain not tired enough where what I'm saying is like, oh yeah, I remember that. I know the level of exhaustion right now can be pretty extreme. So I'm hoping these review sessions will get your neurons firing. Okay, so biological nitrogen fixation is, again, you know, we had an essay question on this in the other exam, but I won't be doing that and I won't be looking for that level of detail. So again, I'm zooming out and looking at the big pictures idea. So the outline that I have there on the Word document, like what is the overall reaction? What are we even talking about in terms of the chemical reaction? And so you don't need to know the details, just the idea that it is N2 going to N2 ammonia, and that the other big picture idea, it takes a lot of ATP. It's not cheap. So N2 to ammonia and 16 ATP, that's costly. Understanding that that is costly. But it must be the benefits outweigh the costs because many plants still stick with this. And then like terminology, what's an infection thread? Like how do they initially invade? It's through the root hairs, you know, and that's where you see the infection thread. And then no membranes are being crossed, just like with the VAM fungi, no membranes get penetrated. And then thinking about what's a nodule, what's going on there. And then of course the enzyme that's driving that reaction, it's peculiar. It's amazingly sensitive to oxygen, which creates problems. So understanding that dinitrogenase enzyme, it has some weaknesses to it, and that it's sensitive to oxygen. But plants and their associated microbes have figured it out. They've adapted a mechanism that, like hemoglobin. So understanding that, like hemoglobin, is important to regulate that oxygen around the bacteria, making sure there's not a bunch of free oxygen floating around where dinitrogenase is. So like hemoglobin is super important. So that's a big picture idea. If that wasn't in play, the enzymes would keep getting inactivated and useless. Questions? All right, let's think about the light reactions then. So again, I'm going to be zooming out. The level of detail will be less. And so, I have on the handout or the outline, certainly where does it take place, knowing it's inside the chloroplast and the chloroplast anatomy. So again, the jargon of the chloroplast, what's going on in the lumen versus stroma, and then you have the thylakoid membrane. So those terms, of course, and then like the big picture, like what's the overall idea here? Why are they even bothering to run the light reaction? So we're thinking about what's the payoff? What are the products that are getting made? And so, and then looking at, well, what's the ultimate source of electrons? Going all the way to the far left of the diagram here, we're thinking about water. And knowing that photosystem 2 is the place for that. So the oxygen evolving complex, photosystem 2, knowing the players, right? So both the big ones, the transmembrane complexes, plus the mobile carriers. And then thinking about what are the mobile carriers doing? Like, where are they acting? Knowing that level of, the PQ is going to photosystem 2 and the cytochrome. PC on the other hand is going to cytochrome to photosystem 1. And then don't forget the paradoxin and FNR, those are technically mobile carriers. Yeah, they're enzymes, but yeah, they're mobile carriers. They're carrying that enzyme, paradoxin gives it to FNR, and that FNR has to deliver it to NADP+. So knowing all the players in the process. But I'm not going to ask, like, I'm not going to say, tell me who all the donors and acceptors are in photosystem 1. I'm not going to ask you intimate details about the Q cycle, other than why bother running the Q cycle? There's a reason plants have evolved that extra little twist to this game that they're playing. So questions on any of that? No questions on the light reactions? Also on the outline, I mentioned antenna complexes, and then the idea of inductive resonance. So again, that's a definition idea. What does that mean? When you look at inductive resonance, it's different than this. Here we're talking about electron transfers going on. Inductive resonance doesn't do that. It's just energy transfer, not electrons. So it's that tuning fork analogy of energy being transferred from one antenna pigment to the other. And it's not really until you get into the reaction center inside the photosynthesis that you actually see electrons getting excited and boosted off, donated off. Any questions on the slide set here? It's a big slide set. Whoops. Here's my PowerPoint. Oh, well, that was not helpful. Exactly. So with the Q cycle, we're looking at it allows for more proton pumping, essentially, is how they typically phrase it. It is causing a situation because, especially because you have the, well, let me put it, the big picture idea here. So this is essentially the Q cycle where the first PQH2 disembarks off of photosystem 2, bumps into the cytochrome B6 up. You get one electron going to that PQ. P2, it's a hod for electrons. It wants 2 electrons. So all of that happens again in order to get 2 electrons to that Q cycle cluster. Like the final is going to grab 2 protons. So not only do you have this mobile carrier dumping and doing it frequently, because it needs, this PQ needs 2 electrons. You have to have that happen twice. It's dumping, and then this PQ is also making protons disappear, just like this one grabbing protons from the stroma. This PQ grabs protons from the stroma. So you see a disappearance from the stroma, dumping into the lumen by both of those PQs in the Q cycle process. So it really ramps up the number of protons that you can get dumped into the lumen, which, why do we care about that? So what? So we get a bunch of protons dumped into the lumen. What's the payoff? It's not really being shown here what the payoff is. ATP synthase. Yep. So they didn't complete the picture on this particular graphic, but it's this idea of that gradient is going to get established now, that proton gradient. And so the protons want to diffuse back based on the law of diffusion, but they can't go through the membrane to go through the ATP synthase. A little molecular motor, and that allows for the rotational torque force gets these 2 molecules, ATP and phosphate, to react. So that's the huge payoff. Proton gradient, anytime you see a proton gradient across a membrane, whether it's a chloroplast, mitochondria, a cell membrane of a bacteria, immediately you should say, oh, that's potential energy. I bet the microbe could use that for ATP, or the plant could use that for ATP. So electron transfer and proton gradients typically go hand in hand, no matter what organism is doing it. It's a common strategy. It's kind of a universal thing. It's a big picture concept, so I definitely will ask about that. It's a huge concept that you'll see that in all your other classes too. Okay, so we'll do part 2 on Friday. Again, bring your laptops so we can work through it, and I'll answer any questions you have.